From Diamond Light Source, this is the Diamond Podcast.
Hello and welcome to the Diamond Light Source Podcast with me, Meera Senthilingam. This month, we're celebrating the International Year of Chemistry, which may be drawing to a close, but we're seizing the opportunity to look at a wide range of chemical discoveries and applications taking place at Diamond. We'll be investigating the role of chemistry across many scientific disciplines, including research into corrosion, new materials for photovoltaics, the synthesis of nanowires for use in molecular electronics, and even identifying a new source of mercury poisoning from preserved plant samples.
Over time, what's been noted is the initial mercury solutions, pretty concentrated mercury solutions, either sprayed on or paintbrushed on, or the samples themselves were dipped into this solution. And what has been happening over time is this mercury chloride has been converted into different forms of mercury, one of which is volatile mercury, which is then being released from the samples.
Manchester University's John Fellows will be revealing how his team have been analysing plant samples to reduce and control exposure to these vapours of mercury later in the programme, when we'll also be bringing you the latest news and events from Diamond. So plenty to look forward to in this December edition of the Diamond Light Source podcast.
The Diamond Podcast. For more information, look us up online at diamond.ac.uk/podcast.
2011 is the International Year of Chemistry, a global celebration of chemistry and the ways research in this field have benefited humankind, with the theme Chemistry, Our Life, Our Future. And one place the path of chemistry is being paved is the Diamond Synchrotron, where everything from designer molecules, pitting corrosion and solar cells are being investigated. Diamond's Director of Physical Sciences, Trevor Raymond, explains the importance of this versatile subject.
As a chemist, by passion and by background over many years, you won't be surprised if I say that I think it's the central science. It's the science that so many other topics interface to, and without which they wouldn't succeed. So I think of it spanning into physics, where high-temperature superconductors came out of a chemistry background, solid-state chemistry. I think of biology. Chemists are the people who actually make the molecules that go to form drugs. There are all sorts of areas, such as catalysis, the clean cars, new sources of energy. In all of those cases, a knowledge of chemistry is vital. And in fact, chemistry makes so many of these things possible. So perhaps you can see that I'm biased, but actually I think it's a great subject and it's central to so many other subjects.
And your own research within all of this is the use of chemistry, I guess, and its role towards the field of materials science.
Yes, I've been working on methods for many years that look at materials under their real operating conditions, looking at their surface chemistry. And what I'm doing at the moment is working with a close colleague in Birmingham, Alison Davenport, to use those skills to look at corroding layers, to look at pitting corrosion, where little black dots in stainless steel corrode. It's quite an important form of corrosion, looking at the chemistry of those corroding pits.
And what have you been able to find out about them, I guess, so far? And is it then the aim to kind of prevent this occurring?
What we've learned is that the chemistry at the bottom of these corroding pits is important. And for the first time, we've identified the chemical compounds that are found at the bottom of a corroding pit. And now what we've discovered, or it's Alison's group has really discovered, is how that chemistry influences the rate of corrosion. Often it's very difficult to prevent corrosion, and so what you really want to understand is how long will it be before it gets really important. And that's where the work is progressing. It's taking the insights that we get from chemistry to predict the lifetime of a device, the lifetime of, example, a canister that might store radioactive waste. The data that we're providing, Alison's group are providing, is allowing us to have really good models of how they might corrode over a period of a decade, 20 years, 10,000 years.
Now, you did mention that chemistry stems into a whole variety of fields, such as biochemistry and physics as well. So what perhaps is some of the more current research taking place here at Diamond that stems into these areas?
I think I'd like to point out two areas. One is the general field of energy. There is, as everyone knows, a real pressing need to reduce the amount of energy that we use and reduce the impact of our industrial processes on the environment that we live in. So if we think about energy itself, then we might think about hydrogen storage. There has been a great deal of work over many years to improve methods for storing hydrogen. And one of the favoured topics at the moment, one of the favoured areas, is the so-called metal organic frameworks. Central to all of this is designing frameworks of molecules that will absorb hydrogen. And it turns out that the synchrotron is the ideal place to determine the structure of these complex materials. On the other hand, there's a lot of interest in designing solar cells. We're all familiar with solar cells now on roofs. They're made of silicon and they're very good, but silicon is an expensive material. It uses quite a lot of energy to process it, and the thought has been, can we make lighter weight, cheaper solar cells made of polymers, plastics? And what matters there is knowing the structure and the behaviour of those layers, which are very thin, studying that and determining that, and synchrotrons are ideal for that.
Now, that work's done by David Lidsey, who we've had on the podcast in the past. And I think an interesting thing with that particular research was that although the efficiency may not be as great as silicon photovoltaics, the fact that they're cheaper and also more flexible and have perhaps a wider array of uses means that they're quite important.
If you're trying to create new energy sources, then ultimately you have to compete with other sources on economic terms, and into that equation comes lots of things like the original cost, the efficiency, ease of installation. So it's really quite complicated, and into an environment like that, having control of the chemistry is really important.
And so you've touched on these areas that chemistry kind of stems into, but what about, I guess, more fundamental chemistry that's looked into here?
Another area that stems out of the control that chemists have over the shape of molecules is the notion that if you take a small molecule that is simple to make, how can you use that? Well, one way of doing it, which is very attractive, is simply to take that small molecule as like a Lego brick and to make it sufficiently clever that it's got bumps and holes in it so that those bricks will automatically assemble if you put them together. That notion of building big molecules from small molecules by specially designed locks and keys, in fact, is the area of supermolecular chemistry.
That was Trevor Raymond, Director of Physical Sciences at Diamond Lightsource. Now, as Trevor mentioned, an exciting area emerging from research at Diamond is the synthesis of new specialist molecules known as supramolecular chemistry. And the scientist pioneering this field is Harry Anderson from the University of Oxford.
I'm interested in organic molecules with strongly coupled electrons and controlling their properties, often using non-covalent, weak, reversible interactions.
And what kinds of molecules are these?
Well, they tend to be coloured compounds, a bit like dyes. A lot of the molecules we work with are made from porphyrins, which are naturally occurring pigments, but the ones we work with are not naturally occurring. I mean, we make them ourselves from scratch, but they're related to the compounds that make blood red and grass green.
And so are you essentially building molecules and then working with them from there?
Yes, we do synthesis. So from very simple starting components, we make compounds and we design the compounds. So we're making compounds that have never existed anywhere before. And so we're interested in learning about their properties and relating the properties to the molecular structure.
But now the actual molecules you're making are quite big. So how do you actually set about, I guess, constructing these from scratch?
It's sort of classical organic synthesis, except that we also use templates quite a lot. So a template is a molecule around which you can build another molecule which determines the shape, holds the components in the right places as you link them together, and then you can remove the template afterwards. So we've developed new ways of using templates to make bigger molecules.
And then is it hoped that they could have some new applications as well?
Yeah, of course. So the sort of molecules we work with have strongly coupled electrons, which means that they're often semiconductors. So organic semiconductors is one important area of applications in devices like, display devices, electroluminescent displays, but also their interaction with light. So they're often fluorescent and have unusual ways of interacting with light that can be used in optical switching devices. And some of the materials we work with generate singlet oxygen, which can be useful in medical applications.
And so could you give perhaps an example of a particular molecule that you've worked on recently? So how you'd set about actually making it, why you wanted to make it, and maybe some of the uses it could now have? One
of the sorts of molecules that we're excited about are molecular wires which are in rings rather than being straight. So we've worked on what we call molecular wires, which are straight molecules down which you can transfer electrons. But recently we've realized that it's possible to make rings of these molecules. So part of the challenge is just to make a molecular wire into a ring and then look at the charge mobility around the ring. But for this particular class of molecules, it's not that easy to identify applications yet. But we think they may have unusual properties, maybe magnetic properties, if we can get them to behave like little loops of metal wire. So we're just investigating them to see how they behave.
What are the applications of the first set of molecular wires then that were created, the linear ones?
Yes, potentially in molecular electronics. That means molecular scale electronics, transistors or electronic devices on the molecular scale, the ultimate in miniaturization as the integrated circuit gets smaller and smaller. So a lot of people are working in that area, but it's still a very long way from being useful. On the other hand, in devices made from on a larger scale, from larger amounts of material, like solar cells and maybe also organic transistors, it's useful to have materials which can transfer charge efficiently. The molecular wires might be components in a material, perhaps in a solar cell.
But would the circular forms have different applications? So you've mentioned that they could alter perhaps magnetic fields and so on. So
yes.
Just changing the shape simply of them could mean give them different properties and other uses.
Yes, just like a bit of wire, if it's in a solenoid shape, behaves quite differently to if it's in a straight shape. That's one of the amazing things about molecules, that just changing the shape and thinking about the shape often relates to the properties.
And how do you use diamond in all of this? So how do you use synchrotron radiation to perhaps aid this process?
For structurally characterizing the compounds that we make, proving that they are actually the compounds that we think they are, and for getting information on the shapes. So there are two techniques that we use at Diamond. Small angle X-ray scattering of solutions is very useful for getting low resolution information on the shapes of the molecules. It hasn't been used much before for synthetic molecules. It's used a lot for proteins. And it's useful because you don't have to grow crystals. You can take a compound and take a dilute solution, and just from the small angle X-ray scattering you can get information on the three-dimensional shape of the molecule. And the other technique that we use at Diamond is X-ray crystallography, and it gives much higher resolution information than the solution phase scattering.
But is there a particular molecule that's been perhaps your most successful to date in terms of how far you've managed to go with it?
Well, I suppose we've worked most on porphyrin-based molecular wires. Perhaps they've been most successful because they're so versatile. So a similar family of molecules can have potential applications in medicine and photodynamic therapy, and also in molecular electronics, and there are lots of other potential applications too. They have such a rich range of properties.
Harry Anderson, Professor of Chemistry at the University of Oxford.
Did you know that Diamond's floor-level monitor system shows Diamond tilting twice a day due to the moon passing overhead?
You're listening to the Diamond Light Source podcast, and this month we're celebrating the International Year of Chemistry and bringing you some of the latest developments in the field taking place at Diamond. Still to come, we discover how researchers at the University of Manchester have identified a new source of exposure to volatile mercury, coming from museum samples. But before that, let's join Sarah Boundy from Diamond's communications team for a roundup of the latest news and events from the Synchrotron, starting with a good reason to drink that cup of coffee in the morning.
Yes, so it seems that coffee could offer a key ingredient for new treatments for Parkinson's disease. And one of our industrial users has used our microfocus macromolecular crystallography beamline, I24, to solve the structure of a protein that's involved in Parkinson's disease and other neurological disorders. The team was from Heptares Therapeutics, and they were looking at the adenosine A2A receptor, which is responsible for regulating the effects of neurotransmitters in the brain, in cardiovascular and immune systems. And they were also looking at how xanthine-based drugs, such as caffeine, bind to their target.
And so what effects did they see? How does it bind?
Well, although it was known that caffeine inhibits the action of the adenosine, the exact molecular mechanism involved was not fully understood until now. So the structural information that Heptares got from I24 has helped them to understand what is happening at the molecular level when the drug binds to its target and blocks the receptor's response. So this information is now enabling them to develop highly optimised next-generation drug candidates for Parkinson's disease and other neurological disorders.
And staying on the field of structural biology, other research taking place here at Diamond was looking into another disease, and that's heart disease.
Yep, so scientists from Imperial College London and Diamond worked at the membrane protein laboratory at Diamond, and their research was recently reported in Nature. They revealed the structure of a cholesterol-lowering drug target. Their findings could hopefully lead to much more effective drugs to tackle high cholesterol levels, which is a big cause of heart disease.
And what is this target and how could it make a difference?
Well, for the first time, they determined the structure of the bacterial homologue of the apical sodium-dependent bile acid transporter protein, or ASBT for short. And ASBT is a target for hypercholesterolemia drugs because it can affect the level of cholesterol in the blood. So there are currently a number of existing ASBT inhibitors effective in animal models, and they were developed without structural knowledge of the protein. Now that they know the shape and the size of the drug binding site within the bacterial model of the protein, it should be possible to work on the design of improved drugs which are much more targeted and which will fit much better.
So two very important diseases of today currently being targeted here.
Yeah, the life sciences makes up about 40% of Diamond's research work, so we're working on a lot of important diseases.
And staying with structural biology still, you had quite a notable scientist in this field visit Diamond recently.
That's right. At our annual user meeting, the keynote address was given by Professor Venki Ramakrishnan, who is the joint winner of the 2009 Nobel Prize in Chemistry. He spoke about his work on the structure of the ribosome and the way discoveries in macromolecular crystallography have accelerated over the past decade, thanks to more powerful beamlines and better detectors and increased automation. So the user meeting saw about 200 scientists from across the UK gather at Diamond to discuss the latest developments in synchrotron science. It was a really successful meeting, and we hope it will lead to much more exciting developments and collaborations between our users.
So that's a benefit for the users, but you've also done a lot lately to benefit the public in terms of the multimedia content that you've got online.
Yep, so Diamond News is hot off the press. The latest issue includes articles on research working to protect herbarium staff from mercury-contaminated specimens. We'll hear about that from John Fellows from the University of Manchester later on. Also looking at the role of gold nanoparticles in cancer treatment, and there's also a feature on polarised light which will help to explain more about our nanoscience beamline IO6 and much more, and copies are available from our website.
In addition to this, there's also visual content, so videos.
That's right, I mentioned them last time, and we've now got a whole host of video case studies available, and you can find them on our YouTube channel. They cover all kinds of things that Diamond is involved in, such as structural biology, industry, innovation and impact, and also environmental and health research. So this gives the viewer great insight into Diamond, but if they actually want to come here and see it in the flesh, our next Inside Diamond Public Open Day is going to be the 14th of January.
Thanks, Sarah. Sarah Bandy from Diamond's communications team, who'll be back in the next edition with more news and events from the light source. But in the meantime, visit diamond.ac.uk for more news and to sign up for the next Inside Diamond Day in January before it books up. Now, moving back to the world of chemistry, we meet Professor Tony Ryan from the University of Sheffield, who's also on Diamond's board of directors, having long had a passion for chemistry and the use of synchrotrons to further this area of science.
So I study the structure of polymers, plastics, and colloid suspensions. So one example might be the structure of the molecules that make up a shampoo bottle, and also the structure of the molecules that make up the shampoo.
And how do you set about looking into this?
Well, we do various things in the lab. So we synthesize special molecules. We synthesize molecules that we can label so we know where they are. And then we use the synchrotron as a source of high-intensity X-ray beams that allows us either to penetrate deep into a sample or to look at a sample on its surface as it's doing its stuff. And we can work out where the atoms are and how the whole molecules are organized.
And what's your real aim here? So what are you actually trying to find out about the molecules?
So if we take the example of a shampoo bottle, then we'd be wanting to know when the material was processed, how the stretching of the molecules affected the way they crystallized. So when you make a bottle, you take a liquid and essentially you blow a bubble in the liquid to a special shape. That's the shape of the bottle. And then the stretching and the cooling causes the polymer molecules to organize themselves so that they crystallize. And we study the stretching and the cooling and the crystallization by following what the molecules do in real time by X-ray scattering.
And what have been perhaps some of your key findings then, having monitored the molecules in this way?
Well, we've just had a paper published that compares the crystallization of synthetic polymers like polyethylene with the crystallization of silk from a silkworm by using the synchrotron. And by using other techniques in the lab at home, we've been able to estimate the energy needed to do the processing. And we find out that the silkworm uses about 10% of the energy that we use to process polyethylene to make a fibre. So silkworms are much better at making fibres than people.
So is the aim then to really find out a bit more about how the silkworm does this to reduce our energy needs? Well,
so what we can do is, from what we've learned about how silkworms go about processing silk, we've learned that it's a different mechanism to the mechanism that's used in making fibres from synthetic polymers. So what we can do is look to mimic the silkworm's method in engineering materials. What the synchrotrons allowed us to do is compare the energetics of the two processes, learn that one process is much less energy intensive than the other, and now we understand both how and why it's less energy intensive to try and replicate that methodology in synthetic materials. So it actually means going back to the molecular drawing board and doing synthetic chemistry again.
Could you perhaps give a bit of insight into an even wider range of areas of chemistry that the synchrotron can be used for and to benefit, say, other scientists?
So my colleagues in the chemistry field use the synchrotron for many, many reasons. So they do spectroscopy that they can't do anywhere else to see what the local environment of individual atoms are, how catalysts work, like the catalytic converter in your car. Other colleagues use the synchrotron to study crystallization of drugs. So you can separate polymorphs, different forms of drugs, because drugs crystallize in different ways, and if you have the wrong sort of crystals in a tablet, they don't work. But they can even work out how proteins interact with each other. So how proteins interact with drugs using a different set of scattering techniques called macromolecular crystallography. So there's a whole variety of different techniques. Imaging techniques, spectroscopy techniques that chemists use to study many, many different aspects of science, whether it be how rain clouds form, how drugs are taken into the marketplace.
And I guess with the opening of new beamlines, which you must know about as well, being on the board of directors and so on, this must just increase as each beamline opens as well.
The new beamline proposals open new areas of science. So, for example, parts of what we do in terms of molecular engineering are being transformed by the availability of microbeams. So you can look in very fine detail in large parts, or you only need very small crystals in order to be able to solve a structure. So all the time the techniques and technology at the synchrotron are pushing the envelope of what we can do, and that generates new scientific opportunities with it.
And what will you be looking into next then? What will be your next use for the synchrotron for your own work?
Well, I'm really interested in how we use catalysts to clean up the environment. So I've been working with an artist on making catalytic clothes. And so what we want to do is we want to stick tiny catalyst particles to people's clothes so they can walk around and take out environmental pollution, to use people and their clothing as catalyst supports. But to do that, we need to understand how the catalyst particles stick to fibres. And so my next project at the Synchrotron will be to understand this process of depositing nanoparticles onto fibres, and we'll use both imaging and scattering techniques to do that.
That's a really interesting idea, and if you can just get a certain percentage of the population to wear that, that could make quite a big difference.
Well, it'd actually have to be quite a large proportion of the population. Each person could take out about 3 grams of nitric oxides a day, which doesn't sound like very much, but once you have 1 million people doing it, then they take out 3 tons of pollution a day. And once you start to get those sorts of numbers of people involved, you can have a significant impact on an urban environment.
So how can people get involved with this and find out more?
So there's a great website called catalyticclothing.org. There's been lots of social media activity around this. At some point, we're going to be doing a pop-up laundry, and we may even run a pop-up laundry at Diamond.
Tony Ryan from the University of Sheffield. Now, that's almost it for this month, but before we go, we've saved one last piece of exciting chemistry for you to hear. And it may just stick in your mind the next time you visit a museum, as it seems some museum samples could be harbouring volatile compounds of mercury. But no need to fear, as it's only the people handling the samples themselves that need to watch out. And to help them, John Fellows from the University of Manchester is on the case at the university's herbarium to stop these vapours in their tracks.
Mercury salts were widely used to preserve museum specimens against damage from bacteria, insects, fungi, and so on and so forth. And you can tell by the state of the preservation that these mercury salts have worked. But over time, what's been noted is the initial mercury solutions. You're talking pretty concentrated mercury solutions. One recipe out there has it as 30 grams per litre mercury chloride dissolved into methylated spirits, which were then either sprayed on or paintbrushed on, or the samples themselves were dipped into this solution. And what has been happening over time is this mercury chloride has been converted into different forms of mercury, one of which is volatile mercury, which is then being released from the samples.
So is the problem then that when people go to work with these samples and open them up, essentially, they're being exposed to these volatile levels of mercury?
Well, that was one of the things we were investigating, yeah. The first thing that we had to test was making sure that there was no dangerous effects in the area. The samples themselves were located in the herbarium, and the first thing we tested was the mercury content of the air in the herbarium, and we found that it was well below the guidelines. We had 1.7 micrograms per cubic metre of mercury when the guidelines were up at over 25.
What were the concerns really with this to mean that you're then obviously analysing these samples, though, to see the levels of mercury within?
The samples themselves may or may not have been contaminated with mercury, because the problem is that over the course of the last several hundred years, because these collections date back hundreds of years, have been kind of homogenized and kind of rearranged and resorted and set out. So they don't know which samples have actually been treated. And the boxes that these samples are kept in are these little airtight containers, or little cardboard boxes, which are essentially airtight. And what we were mostly concerned with is like this build-up of mercury within these boxes, which then, if someone could open the case, and then you'd have this kind of waft of mercury, which overall across the entire area is not a significant concentration, but it's that initial kind of opening the box that we wanted to look at.
So how have you set about doing that?
Okay, so, I mean, the other aspect we were looking at is, part of my work was, as a biogeochemist really, is to analyse how efficient our biogenic selenium nanoparticles are. That's selenium nanoparticles which have been precipitated by bacteria. And one thing that's been published out in the literature is that selenium is very, very good at capturing this elemental mercury phase. So it was kind of a twofold research in that, one, we wanted to identify and quantify any mercury present within the samples, and secondly, to find a way if we could test our selenium nanoparticles and how good they were at capturing this mercury.
How have you gone about doing that?
What we made is little filter membranes. Our selenium starts off in as a suspension, and you can filter them out onto these filter membranes, which are these 0.22 micron filters, and then allow them to air dry. And we placed them in some of the boxes where we knew samples had been contaminated with mercury, and see if we could capture the mercury as it was coming off the samples.
So I guess essentially what? You would be making these membranes to soak up the mercury just prior to opening up samples, or how would this process work if these are proven to be effective at soaking it up?
The samples themselves we'd be leaving inside the boxes permanently, so that hopefully would decrease any build-up of mercury within the boxes.
And so how have you been using Diamond to try and, I guess, visualise this?
Yeah, Diamond was crucial, really, because we needed to investigate the form of the mercury present, which will help us to understand the fate of what happens to the mercury, but also because these samples are so varied. I mean, some have been contaminated with mercury, some haven't, and some of the samples which have been contaminated are only contaminated in areas of the sample. So Diamond, to start with for the samples, was helping us to pinpoint where the mercury was and also tell us the kind of concentration we can expect and the form that it's in. Secondly, it also helped us to characterise the phase the mercury takes once it's been reacted with the selenium and ensure that it's not about to come back off again.
And what were some of your key findings then? So how badly contaminated were the samples?
So within the herbarium, there's numerous collections, and the one we focused on was the European collection. And once we'd identified a sample that had got the mercury, we needed to find out whereabouts it was. What we found is that the mercury itself tended to be as little crystalline mercury sulfide phase as it happens, which was Diamond that helped us prove this. There's one particular sample which was really quite good, in that it's a collection of dust from the bottom of one of these boxes. So just to put this in context, the samples were stuck down onto A3 sheets, and these A3 sheets were then piled one on top of the other within these cardboard boxes. And what we found when we were looking through was we were trying to identify where this mercury is, and none of the sheets really came very high on our mercury, on the scale of mercury. But when we analysed one of them, we found that the dust which had collected on the bottom of the box, so none of the ones on the sheets, but the actual dust at the bottom, when we analysed that, we found quite a high mercury concentration, which we picked out by XPS, and then we sent off to Diamond for XAS. And what we found on this is maybe a micron-long particle contained on one of the dust particles, which was almost pure mercury sulfide. Once we analysed these samples in Diamond, they were able to scan across our sample set and pick out, saying, right, there's a bright bit right there, let's go in and let's get our XAS spectrum from this little sample here.
Now that you've been able to see this spread and localisation of mercury within this variety of samples, what are you able to do with the information, I guess? So what do these findings really mean?
So what we can do with this data now is we can kind of help build up health and safety guidelines as to what people are to do with these samples. What this has helped build up is saying, right, okay, what you want to do when you find one of these boxes is open the box and then just leave for 5 minutes and then come back and then go through the samples. Because it's the elemental mercury phase, the volatile phase, which is the dangerous one you want to watch out for. The mercury sulfide that's going to be in there is not really too much concern. We'd like to get to the stage where these selenium membrane filters are used within the boxes to help prevent this build-up of mercury within the boxes, so that when someone comes along to look at these samples, they can lift off the lid and not have to worry about a waft of mercury coming out from the box.
So before these membranes are in use, mental note, leave the room when opening a museum sample. Well, some of them anyway. That was John Fellows from the University of Manchester. Now that's it for this edition of the Diamond Podcast, but do join us again in the new year, as 2012 will be bringing you more insight into the many areas of scientific research taking place at Diamond, including Diamond's 10-year anniversary celebrations. In the meantime, if you have any questions about Diamond or the research taking place there, email podcast@diamond.ac.uk. You can also listen to previous editions of this programme online at diamond.ac.uk/podcast or thenakedscientist.com/diamond. You can also subscribe to the podcast on iTunes. Thank you this month to Trevor Raymond, Harry Anderson, Sarah Boundy, Tony Ryan and John Fellows. I'm Mira Senthilingam. Thank you for listening, have a lovely festive season and see you next year.
The Diamond Podcast is brought to you by Diamond Light Source and produced by thenakedscientists.com. There's more information on our website at diamond.ac.uk/podcast.